Bismillah ar-Rahman ar-Rahim. Today, inshallah, I'll talk about glycogen metabolism. Continuous supply of blood glucose is an absolute requirement for a human life, as glucose is the sole fuel for the human brain. Glucose is also required for or as an energy source for cells with a few or no mitochondria, such as mature erythrocytes. It is also an energy source for exercising muscle during anaerobic glycolysis. Blood glucose can be obtained from diet, but dietary intake of glucose and its precursors is sporadic, so depending on diet is not always a reliable source for blood glucose. Gluconeogenesis is another source of glucose which can provide sustained synthesis, but a problem with gluconeogenesis هي إنه it's slow in responding to a falling blood glucose level. Therefore, the body has developed mechanisms for storing a supply of glucose in a rapidly mobilizable form called glycogen. So, what is a glycogen? Now, if we break the word glycogen, the prefix gluco means glucose and the suffix gen means producing. So, degradation of glycogen will produce glucose when it is needed. Glycogen can be defined as a branch-chain polysaccharide, a polymer of glucose monomers. Glycogen is synthesized and stored mainly in liver and in muscles, although most other cells store small amounts of it for their own use. In liver, glycogen is converted to glucose 6-phosphate and then to glucose and then released to the blood. Function is to maintain blood glucose concentration, while in muscles, glycogen is degraded into glucose 6-phosphate, which is used as an energy during muscle contraction. Here it's not changed into glucose because we don't have glucose 6-phosphatase. The enzyme glucose 6-phosphatase is found mainly in liver and in kidneys. Sometimes it's also found in the small intestine. Glycogen is stored in cytoplasm as granules. Its stores in liver increase during the well-fed state and are depleted during a fast, while muscle glycogen is not affected by short periods of fasting. It might moderately decrease in prolonged fasting. Structure of glycogen. This figure shows us the generalized structure of glycogen. As I said, glycogen is a branched chain polysaccharide, a polymer of glucose monomers, particularly alpha-D-glucose. In the linear form, we have alpha-1,4 glycosidic bond, means that the first carbon of first glucose is attached to the fourth carbon of second glucose, while at branching point, after an average of 8 to 10 glucose residues, there is an alpha-1,6 glycosidic bond. Again, glycogen exists in discrete cytoplasmic granules that also contain most of the enzymes necessary for its synthesis and degradation. Glycogen synthesis takes place in the cytosol, and as a metabolic pathway, glycogen synthesis needs a monomer of UDP glucose. It can't use glucose as it has to be covalently linked to the uridine diphosphate. Glycogen synthesis also requires ATP for glucose phosphorylation. So glycogen synthesis requirements are ATP, uridine triphosphate, UTP, and the basic substrate for the whole process, glucose. Now let us talk about synthesis of glycogen step by step. Synthesis of glycogen requires the coordinated action of a number of enzymes. Glucose enters the cells via special glucose transporters, being phosphorylated to glucose 6-phosphate by hexokinase isozymes using ATP. The next step is the isomerization of glucose 6-phosphate to glucose 1-phosphate via phosphoglucomutase. This enzyme, I mean the phosphoglucomutase, transfer the phosphate group from C6 of glucose 6-phosphate to C1, converting it to glucose 1-phosphate. Phosphoglucomutase transfers phosphate from the 6th carbon to the 1st carbon. At the first, the phosphate group of phosphoglucomutase will be added to the 1st carbon of glucose 6-phosphate, making glucose 1,6-bisphosphate as an intermediate. Then the phosphate on the 6th carbon will be taken by the same enzyme, thus making glucose 1-phosphate. This enzyme is the bidirectional enzyme, plays a role in glycogenolysis in addition to glycogen synthesis. After glucose 1-phosphate is made, 1-uridine triphosphate UTP will join to form UDP uridine diphosphate glucose by the enzyme UDP glucose pyrophosphorylase. And here, UDP glucose will be the first product of the reaction. Pyrophosphate will be released as a second product of the reaction. This is a high-energy compound with 2 phosphate groups, will be hydrolyzed, giving 2 inorganic phosphates by the enzyme pyrophosphatase. And this ensures that UDP glucose pyrophosphorylase reaction proceeds in the direction of UDP glucose production. So now we have UDP glucose ready for glycogen synthesis. But glycogen synthesis, the enzyme responsible for making alpha-1,4 linkage between glucose molecules in glycogen chain, cannot start from scratch. This enzyme cannot initiate chain using UDP glucose. Rather, it requires a short sequence of at least 4 glucose molecules attached together. This short sequence is known as a glycogen primer. From where do we get this primer? Now, in cells whose glycogen stores are not totally depleted, a fragment of glycogen can serve as a primer. What happens if we don't have this glycogen primer? If stores of glycogen are totally depleted? We have a protein called glycogenin. This protein has tyrosine amino acid, works as an acceptor of glucose residues from UDP glucose. As you can see in the figure, glycogenin transfers a glucose residue from UDP glucose to the hydroxyl group of its own tyrosine. The side chain hydroxyl group of a specific tyrosine serves as the site at which the initial glucosyl unit is attached. This reaction is done by glycogenin itself via autoglucosylation. That's why glycogenin is considered as an enzyme. Glycogenin then catalyzes the transfer of next glucose molecules from UDP glucose, producing a short alpha-1,4 linked glucosyl chain, which serves as a primer. So once we have 4 glucose molecules bonded to the glycogen synthase enzyme can act. And here we have to know that glycogenin stays associated with and forms the core of a glycogen granule. Elongation of the chain involves the transfer of glucose from UDP glucose by glycogen synthase releasing UDP to the non-reducing end of the growing chain, elongating it. The bond here between glucose molecules is alpha-1,4 bond or glycosidic bond. The UDP released when the new alpha-1,4 glycosidic bond is made can be converted back to UDP by nucleoside diphosphate kinase enzyme. An enzyme called branching enzyme 4,6-transferase take about 6 to 8 glucose from non-reducing end, add it to any glucose in the chain. This will transfer the glycosidic bond alpha 1,4 to alpha 1,6 linkage, forming a branch in the glycogen chain. This figure summarizes what have been discussed above, starting from glucose 6-phosphate, which will be isomerized to glucose 1-phosphate via the enzyme phosphogluconeutase. Once glucose 1-phosphate is made, a molecule of uridine triphosphate UTP will join to form UDP glucose via the enzyme UDP glucose pyrophosphorylase. UDP glucose here is the first product of the reaction. Pyrophosphate is the second product. This product has high energy. It will be hydrolyzed via pyrophosphatase to give to inorganic phosphate. Glycogen transfers a glycosyl residue from UDP glucose to the hydroxyl group of its own tyrosine, releasing UDP. Further addition of glucose residues will form a short primer of at least 4 glucose molecules. Glycogen synthase then will act by taking glucose from uridine diphosphate glucose to elongate the chain of glycogen. طبعا الجلوكوز here is added to the non-reducing end of the growing chain, elongating it, and the bonds here are alpha 1,4 glycosidic bonds. We have an enzyme called a branching enzyme 4,6 transferase. This enzyme will take about 6 to 8 glucose from non-reducing end and then add them to any glucose, forming a branch. At the branching point, the bond will be alpha 1,6 glycosidic bond. And by these steps, we will end up finally with a glycogen molecule. That's all for our lecture today. Next time, inshallah, we will talk about glycogen degradation and regulation of glycogen synthesis and degradation. Thank you.